All right, welcome to LAMA2 research session. So I'll be moderating this session. My name is Carsten Bonnemann. I'm a pediatric neurologist, physician scientist at the NIH, NINDS, which is 2 hours south of here in Washington, or close to Washington. Well, 2 hours by train, 4 hours by car, depending on traffic. I'm involved in clinical and molecular research of early onset muscle disease, congenital muscular dystrophies, and LAMA2 clinically, but not in the lab. But we have many experts here that are going to guide us through this. So my role in this is to be the moderator. I will be asking everyone to introduce themselves and just briefly describe the research they're doing. I'll be highlighting some aspects that are relevant to as kind of principles, and then we open it up for discussion, and I'll be a little bit your voice as well, so I'm going to, if you don't challenge them, I will. So just as a preface, so this form of muscle disease is called congenital muscular dystrophy, meaning this is a muscle disease that starts before birth and is recognizable in a newborn or early infancy. The challenges we face with LAMA2 congenital muscular dystrophy are exactly that this is a disease that is early established, that is present when the child is born already. So it's an established muscle disease there that we have to take into account when we think about how we address it and how we measure what we do as outcome measures. The second problem in this disease is that it is, and I don't want to put problems here to kind of discourage you, just to kind of put out the challenges so that we can address them as the panel. The second challenge is that when we look at early muscle in children with LAMA2 muscular dystrophy, there's a lot already happening in the muscle, a lot of muscle turnover and inflammation that we need with our therapies to address and turn over. The next thing to take into consideration is that muscle is actually a large organ in the body. If you think about a human being, depending on your level of fitness and your body mass index, 50 to 60% of your body is muscle. And it's not only one muscle, it's 650 different muscles that all have functions in the body, from movement to expression to vital functions such as breathing. And those all have the muscle disease that we need to address. And then lastly, in congenital muscular dystrophy of the LAMA2 type, we also know that other parts of the body can be affected, in particular the peripheral nerve, and that is particularly relevant in the mouse, and we're not quite sure how much relevance that has in a human being, but we still have to address that. And then also some involvement of the central nervous system, of the brain, of the white matter, but also sometimes of how the brain is formed. So those are challenges that we need to take into account when we think about therapeutic approaches, not to be spooked by them, but to be kind of enticed to do as good as you can to address this disease. So that just as a background, and then I will now ask our panelists to introduce themselves and to talk a little bit about the research. Start with you, Veronica.
Thank you. Do you mind to just hold it? Yeah, thank you. Hi, I'm Veronica. And I'm here both as an affected individual and as a researcher. I'm a biologist, and I'm doing a project regarding genetic modifiers that can actually alter the severity of the diseases affected individuals. I trained originally in the laboratory of Professor Montoni in London, and I recently moved to Milan and Ospedale San Raffaele with Professor Previtali.
Thank you, Veronica. And so here, it's great to have, of course, you in the field. But here's an important concept that is really already evident. That the same genetic disease plays out differently in different people. So sometimes you see it more severe, and sometimes you see it quite a bit milder. And really, that's because the genetic background of every individual is different where the disease plays out. And you really want to know why the people in which the disease plays out milder, why is that? What's different in their genetic makeup that protects them from the muscle disease? Because if we know, then we have a therapeutic target. Maybe we can replicate that in others as well. So that's a really, really important concept. So the next challenge that I didn't mention is that size of the gene. LAMA2 is so big that it doesn't fit into AAV vectors, gene therapy vectors. But then Judith comes in.
Yeah, so my name is Judith Reinhard. I'm a researcher in Switzerland. And I study mice which have LAMA2-related muscular dystrophy since almost 10 years. And we use these mice actually to learn about the disease mechanism and also to test potential therapies. So I've seen many mice which have LAMA2-related muscular dystrophy. Some years ago, I came the first time to the Cure CMD meeting, and where I actually saw the first patient which has a LAMA2-related muscular dystrophy. So we use the mice to study the disease, but we also try to develop a gene therapy for LAMA2-related muscular dystrophy. And since we cannot bring this LAMA2 gene via AAVs into the muscle, we developed some linker proteins which are small enough to be developed, to be delivered via AAVs, and we are testing this therapy now in the mouse models.
So here's another important concept, which is to kind of take advantage of what nature already kind of offers to us, how we can maybe circumvent something that's not properly working because there's a genetic defect that doesn't allow the protein to be formed. And so these linker proteins are designer proteins made from what the body actually offers to us in terms of the linker activity. So there are really re-engineering of nature's own proteins to take up the role of something that we cannot deliver easily. Maybe I should ask Markus to also comment on this because they work together.
Yeah, my name is Markus Ruegg. I'm the old version of Judith. I work as a professor at the University of Basel since many years, and we have developed, before Judith came to the lab, actually the idea of linker proteins, having one linker which we studied as a normal protein in the mice, and then developed this and saw that in this mouse model for LAMA2-related muscular dystrophy, we really got a good improvement in the disease course, and now we have a second linker. And when I presented this 20 years ago, I should say, to the research community, people were asking me, but how do you want to apply that to patients? And I always said gene therapy, and 20 years back, people were laughing at me and saying gene therapy is a no-go, and there is no way that we can deliver this. And I think now we have entered the time that we can actually do this, and this is what we hope for, and that's what we try to do in the lab now.
Great, yes, and we come back to that. Carlos.
Yeah, well, thank you for inviting us today. Yeah, my name is Carlos. I'm a postdoctoral researcher in Markus Ruegg Lab, which is located in Barcelona, in Pompeu Fabra University. And what we are trying to do somehow is developing gene replacement therapy by, as Carsten mentioned it, so the bottleneck of the whole LAMA2 gene is that it's huge to be put into a single vector. So we came up with two different strategies, sort of saying. One of them is actually splitting the whole gene into 3 different parts, which then we can actually assemble back when they are inside the cell. So as one of the main approach, or the most advanced approach that we have actually in the lab is dividing this gene into the 3 parts, creating viral vectors that contain these 3 parts, and then doing the administration. We have tested this on mice, and it seems to work decently well so far. So this is the viral approach that we have, and then we have another approach which is using, I'm pretty sure that some of, or most of you have already heard about lipid nanoparticles due to the COVID-19 vaccination and everything that came with it. So we tried to develop a way of producing these this whole genomic structure, just genomic sequence, and putting it into a lipid nanoparticle that we produced in-house. So by doing so, we avoid this splitting part of the gene, and then therefore once we have these lipid nanoparticles loaded with this payload, which is the the LAMA2 payload, we inject it into mice. And we've seen that early results show that, yeah, it's a possible way to move forward towards what we understand to be a gene replacement therapy.
Thank you. So two important concepts here. One is how to get around the size limitation of AAV. We heard about the linker approach, and just as a backup, AAV is a naturally occurring virus that is not pathogenic, doesn't cause disease in humans, but it's kind of prevalent in many populations. And it actually it comes along with big viruses that cause disease, like adenovirus or CMV. So it is dependent on bigger viruses because it has an incomplete genetic information itself. But what disease researchers figured out early on is you can take advantage of the non-pathogenic nature of that virus by using just the envelope of the virus. So an envelope is literally a packaging capability. It's a bunch of proteins that form an envelope in which normally the virus has its own genome to go along with others to infect and replicate. You can replace the virus genome with something that you want to deliver and take the virus envelope as your tool to get it somewhere, and it will infect only once. It can't replicate because it has no longer any genes in it that would allow it to do so. It's just a delivery tool. Problem is, AAV is small, which is great for production, is not good if you want to deliver big things, such as a LAMA2 gene. So one approach around it, you heard about the linkers, the small linkers that could kind of take the place of LAMA2, and you can package them into separate AAV envelopes. And the other approach is that to deliver the big gene in bits and pieces in 3, sometimes even more AAVs, and ask them to recombine to form the real thing after delivery. Now, that's something that's been developed also for Duchenne muscular dystrophy, where we also have a message that is too big for AAV. It's kind of the curse in muscle disease. Our genes are so big that we often have to kind of work around it. So that kind of splitting up things in smaller units and package them in AAV is one thing. And the other way is to find a different delivery tool, no longer AAV, but something that really can accommodate the full message. And the lipid nanoparticles are one way to do that. The problem with lipid nanoparticles is it's very tough to get them beyond the liver. So when you do gene therapy and you want to get to all muscles of the body, the only way to do that is to inject into the vein and then have it distributed by the arteries to all muscles. But to get to the heart, you have to get to the liver first. And this liver, the liver is very good at soaking up anything that passes through. That's what it does. That's its job. And lipid nanoparticles get to the liver very effectively. There was even a gene editing therapy here developed here in Philadelphia, where they injected it into the vein, go to the liver, edit the liver effectively. But it's very poor going beyond the liver where we need it to be. So we have to be kind of working on two fronts. Work with what's working right now, and AAV is working right now. We'll talk about that later. But at the same time, think forward into the future, what the next step would be. Hailey.
Hi, my name is Hailey Hermann. I'm a postdoctoral researcher at the University of Nevada, Reno. I work in Dr. Dean Birkin's lab. And so rather than gene therapy, we work on protein replacement therapy with laminin 111. This is the embryonic version of laminin 211. Currently, I've been using some advanced technologies called spatial proteomics to study cell signaling pathways in human and mouse tissue. As well as I'm currently doing experiments trying to focus on the systemic delivery of laminin 111, because it is a difficult protein to have run through your body systemically.
So another important concept here, which is the body's own replacement genes. That's something that occurs in also Duchenne muscular dystrophy. There's a backup gene called utrophin that can take the place of dystrophin if you only get it where it should be and is no longer there. And so LAMA1 or laminin, form of laminin 111, the LAMA alpha 1 chain of laminin, has many aspects of laminin alpha 2, of LAMA2, but it's no longer expressed where you need it after birth. So it is no longer there where it could take the place. And so one way to get it there is to develop this as a protein and give it and see whether it gets to the muscle and take the place of LAMA2. And another approach that I don't think we have represented here is to upregulate the body's own backup gene with tricks and ways to see whether you can tune it up so it appears again in the muscle where it belongs and could take the place of LAMA2. And maybe in the discussion we can get to that and discuss it. Avnika.
Hi, I'm Avnika Ruparelia. I'm from University of Melbourne in Australia. So I'm a muscle biologist, very much interested in modeling muscle diseases and muscle wasting conditions, including LAMA2 congenital muscular dystrophy. We particularly use preclinical models, and my expertise in particular is using zebrafish models, so fish models. We can discuss why fish, very happy to talk about that. But using this model, we've got two arms of research, functional genomics, and so quite often a patient will go into the clinic and you're diagnosed and you're given a mutation that this is what we think may be causing your disease. But there's nothing known about that. So in the lab, we can do some cool tests to determine if that novel variant that the clinician has told you, is that truly disease causing? And we can try and help you elucidate, is that variant is what the problem is. Beyond that, of course, then mechanistic insight. So what's going wrong? If you can identify what's going wrong beyond the obvious, that you lack laminin 211, can we get some downstream mechanisms to then therapeutically target them? And that's something that we're working on as well.
Yeah, thank you. So again, two important concepts. One is what models do we have to model the human disease so that we can test mechanisms and therapies. So you heard about the mouse models already, and in LAMA2, that's one kind of blessing, if you will, that the field has. There's plenty of mouse models that are actually very good models of the human disease. For many other muscle diseases, the mouse models are not that great. The mouse, you have to really look very hard to see that the mouse actually has a muscle disease. Not so in LAMA2. There are many different models that model different severities of the disease. There are differences in what is involved. So the mouse appears to have more nerve involvement than the human has. But the severity and what you have to address is really well modeled by the mouse. And that's important because if you can help this mouse model with your therapy, you have to move quite a bit of the disease to show that. And that's different from Duchenne mouse models, for instance, where the disease is much milder. And it's actually, quote unquote, easier to treat the mouse. Here we have a good model. But there are other models like the zebrafish, the cells that we can do, and so there's a really good toolbox out there to study this disease and the therapies. The second important concept is that of mechanisms. So thus far we've talked about addressing the gene itself, the genetic defect, but then after the genetic defect, there's something that happened in the muscle cells that make them actually disappear and have the disease. And then after that, of course, there's a patient with all the medical problems that need to be addressed. And so whenever we think about therapies for a disease like this, we have to think at all levels at the same time, the genetic correction, but also understand what actually happened in the muscle. What can we do to make the muscle more resistant to the disease? What can we do to help the patient deal with the disease and be as good as it can be medically to respond to the therapies? And that's where Anne comes in.
Yeah, that's a perfect segue. So I'm Anne Conley. I'm at Nationwide Children's Hospital in St. Louis, in Columbus, Ohio. I was St. Louis for about 30 years, but now 6 years in Columbus. And in collaboration with Carsten and also Reghan Foley, the NIH funded or put out an initiative to try to be what is called clinical trial ready for this disorder about 4 years ago, actually. It was, and in order to do that, the request was to get, it's a rare disease. I think many of you were at the last meeting where we talked about that somebody's going to see this about every 3 years, even at a big medical center. So the idea was to get 13 sites across the United States clinical trial ready to look at the very youngest children. Carsten and Reghan had done already beautiful work in the kind of slightly older age group, 5 and older. So this initiative was to try to be clinical trial ready for 0 to 5. And so takes a while to set that all up, but at this point, almost every site is up and enrolling, and the idea is to see what kinds of things we can measure in the very youngest children. In an older person, you can measure power or strength or walking, but in the infants and the very young children, we have to be able to measure other things, function, development, and all of those things are being measured. And I'm really both proud and, well, I won't say the other word. I'm proud to be able to be collaborating with Carsten and Reghan as co-eyes on this initiative, which took
So that's a very important concept here as well. This concept of clinical trial readiness and outcome measures may seem, why should we do that if all this exciting work is going on in the lab? It will not work without this preparation. Not only on the level of really showing whether something works, doesn't work, or make things worse, hopefully not, but also in order to be of interest for any sponsor of clinical trials. So when you try to entice like a biopharmaceutical company or even a public granting mechanism to fund a clinical trial, they want to know whether you have the numbers and you know what you're doing. When you test this therapy in children, they want to know whether you actually have the tools in hand to show whether the therapy is actually working with a reasonable amount of effort and patience. So can't overemphasize how important that is to be at this point while everything is in the pipeline and inching towards clinical translation to be ready for that step and not behind, be ahead of the curve. So that's really important. Bert.
My name is Bert Smeets. I'm a professor at the University of Maastricht in the Netherlands. I'm a biologist by training. I work in the field of genetics already for 40 years, and I'm very much impressed in what we achieved by making diagnosis, but very much disappointed in how they led to finding a treatment for any of the muscular diseases so far. So our approach is on stem cell therapy. So at some point we thought, well, if patients would have healthy stem cells, that would also be a possibility to cure their muscle diseases. And we work, as I said, on a number of muscle diseases, and one of them, they make healthy stem cells by themselves. And that is for us a disease where we can really try the treatment to see if it's effective or not. And there we're already in a phase 2 efficacy trial, so we show that it's safe. We administer these stem cells through the arteries and then they reach their muscles. And what Carsten also mentioned, to get funding for these trials is quite important. We are so far able to do that with public funding, so it means that any benefit of the treatment doesn't have to be paid back to investors or whatsoever. So it's also a possibility to keep these treatments for these rare diseases at a reasonable cost. And that maybe Carsten can comment on that later. For LAMA2, yeah, then obviously these patients do not have healthy stem cells, so we are looking at ways to correct them. So that's part of the work we're doing to see if we can correct them. And in the Netherlands, we have a bit of a specific mutation, a founder mutation, which is a splice site mutation, which Carsten will explain in a moment how we can handle those. You can handle those with antisense technologies. Apparently, these patients are less severely affected than the patients who have no protein at all. And it means that you have a way of manipulating the level of LAMA2 they make. They make a little bit of functional LAMA2. So either you increase everything, or we can specifically make that part of that transcript, which is functional, better in a larger amount than, and therefore we can use antisense oligonucleotides. And the delivery issue has been mentioned. The muscle stem cells you can also use as a delivery agent, because that's the natural way where your muscles recover. So they can make new fibers, but they can also release their contents in the existing fibers. And that's things we try to increase. And we do that also by making these muscle stem cells stronger by microRNA cocktails. I give you a lot of challenges to explain in a moment, Carsten. By microRNA cocktails, which can also increase the myogenesis property, but also reduce the fibrosis, because that's also a clear problem in these diseases, that the muscles being replaced by fibrotic tissues, and then which would limit all kind of treatments. That's my short introduction.
Yeah, so here we have another very interesting concept which exists in other genetic diseases as well, which is to take the body's own stem cells, regenerative cells, and correct them and give them back to the body. So that is something that is well established now in certain blood diseases, in some neurological diseases, but remains a challenge in muscle disease because of the delivery that we have to face, where we have to get as many muscles as possible. These cells can be manipulated in many ways. They can be made, they can be corrected, but they can also be used as factories to provide support molecules to muscle that is trying to regenerate. So you can address two things with one delivery tool, a new corrected version of the gene, but also factors that would help the muscle to regenerate and these cells to survive. That is a concept of cell therapies. And so, of course, first steps will be proof of principle steps. So you do it in a very regional way, where you just kind of look at one muscle to see whether you actually make a difference in that muscle, and then address the body-wide delivery later. And so we're going to challenge you on that later. But so here we have basically yet another delivery tool, which is actually a corrected cell. Maria.
Hi, I'm Maria. I work in Vall-d'Hebron Research Institute in Barcelona. And what we do is something similar. So since we know that laminin 2-1 can travel through the bloodstream somehow, we thought about some kind of protein pump or laminin alpha 2 pump that didn't necessarily to be in the muscle or targeting all the muscles, but it could be somewhere in the body that would be producing the protein. So in this way, we thought about mesenchymal-derived cells and tissues. It is not that they naturally express laminin alpha 2 and beta 1 and gamma 1 that are necessary to produce the laminin 2 and 1. And we are trying to use both mesenchymal stem cells and also other derived tissues like adipose tissues. So we know that adipose tissues is so persistent in the body, and we would want to try. So we are trying to use some kind of adipose tissue transplant to produce some from a. Now we are working with animal models, so from one wild-type mice that is healthy into the LAMA2 mouse model, and see if that pump of laminin alpha 2 would be enough to restore the muscle function. And also on the other hand, we have the mesenchymal stem cells that has the different benefits that Bert was mentioning about the stem cellness. It could modulate also inflammatory factors and modulate fibrosis somehow, and also be a pump of the protein. We are also trying to modify them to produce more laminin alpha 2, since we don't know which is the threshold of the protein that we need to have a therapeutic effect.
So yet another concept here that combines some of the approaches that you've already heard with the protein therapy, and the stem cells. And so here we're taking advantage of something that also is already in the clinic for other things, such as clotting disorders, where you use a different cell to produce the protein and then have it delivered with the body's own vascular system to the organs where you want to go. So you basically create a reservoir of cells that would make the protein, but it's not in muscle yet. It would have to be it's secreted into the bloodstream and then delivered to the muscle. So you have basically your protein factory now in the body, but the protein has to get to the muscles to be effective. So it's kind of an internal protein therapy, if you will. So lots of exciting concepts here. Before I will challenge the panel, I give you the opportunity to ask questions of, because this is fairly high level and somewhat arcane stuff, so please ask questions. I will do it for you if you don't. Yes.
Thank you. My question is just around, I think it's this one here, what steps do we still need to complete to become clinical trial ready, and timeline, like what does that look like right now?
Thank you, it's an excellent question. So we'll start with Anne to to ask her where do we stand with clinical trial readiness, and then I'd let some of the preclinical developers say how far they feel they are from translation and what their roadblocks are and what is needed to move into Anne's patient population to do the trial.
So the trial the goal is to enroll 44 children under the age of 5 and to have 22 of them be enrolled prior to the age of 2.5. The reason for all of that is that this muscular dystrophy and most are slowly progressive. So the more we know about the earliest time points, the better. So on that path, and this is in the United States, there's beautiful work in Spain and in Brazil. There's other natural history studies that may be a little further along, but getting this youngest population is the goal of our research. And with regard to how far into that we expect, and I am an optimist, but I do expect to enroll in the next year. We just started enrollment, so in the first month we enrolled 3. But there are at least 24 to 30 others that I believe will be enrolled within the next 6 months. And then it will be new diagnoses at that point. But we've, in addition to the 13 sites, we've reached out to our other colleagues at multiple other sites. So I'm optimistic that we'll hit the 44 as the goal. So once they're enrolled, the idea is to follow each child 2 years every 6 months. And the good news for this trial is that if suddenly something becomes available, we do not say, oh no, don't try that, just stay with our good. No, the exit, so even with 1 year of data on 44, that will be extremely valuable data. And so we might get half the children with 1 year and half the children with 2 years, depending on all this amazing work that you've been hearing about.
Yeah, thank you. Clara, you want to add to that? Or?
Sorry, I asked too much as well. I would like to know, for example, they're using givinostat, and it's a form of improving the change in the muscle to fibrosis and inflammation, and it works for DMD, apparently. Does the same idea works for LAMA2, for example? Is that something that people is looking forward to try?
Yeah, so there are other compounds which interfere with fibrosis. Losartan is one of these, has been tested in the mouse models and also has some treatment effect. So just based on the mechanism, I would think that reducing fibrosis could bring also benefits for LAMA2 MD, but this has not been evaluated in detail.
Yeah, and we come back to that question when we talk about multimodal therapies and how you get them to the clinic. But let me come back to the second part of that question for the preclinical developer, whoever feels like they could speak about it. Where do you think you stand for the preclinical development? Where do you think the roadblocks are?
Yeah, I can. So we are, in terms of preclinical development in the mouse, I think we are quite progressed. So we have, when we treat the mice, we have a very strong treatment effect. But so we are developing a gene therapy. You may have heard about gene therapies before. So gene therapies have high chance of bringing large benefits, but gene therapies also come with a very high risk. So you may have heard recently there were some tragic cases in other muscular dystrophy when gene therapies have been applied. So that's why we have to learn more about, the safety of the therapy. And only when we have tested this and know this in detail, we can move from the mouse to the patients.
Markus, you want to?
Since I'm similar age as Anne, I'm also very optimistic about things. And what we basically need for our linker therapy is production of AAVs, which then express those linkers. We need to do toxicology. We have to test whether these linkers are eventually toxic for rats or non-human primates. And then we also need, of course, a production pipeline. We actually learned, since we are not really gene therapy AAV producers, that AAV production is still in its infancy, I would say. I mean, it's not really a platform that you can produce AAVs. It could be that your linker, your transgene, makes it very difficult to produce sufficient AAVs. We don't know this yet, but we are in the process to move forward, and we hope that we can progress, and in 2 years or so that we can actually, that we know that we can basically produce these AAVs. That's what we hope for.
And how do you do that as a professor of cell biology, and you want to kind of produce something for medical use? How do you do that?
We have created a small company called Seal Therapeutics that actually helps. We have patents for these linkers, that it's important to actually find investors, and we are trying to now raise funds, because this is all not cheap. You can imagine producing these AAVs costs actually quite some money, and we don't have that money yet. And this is what we do. We're using a vehicle, like a small company, to actually do this. We have investors, actually patient organizations from Switzerland invested already money into our company, 1 million Swiss francs. But we need, of course, now about 10 million to actually push this now to the next step.
And what has the response been from the big guys?
That's actually, we were anticipating, since, as Judith has alluded, the preclinical data look pretty convincing. We were hoping that big pharma would eventually pick up on our therapy and then say we step in and produce this, because, I mean, Novartis, which is just next door to us, they have bought this spinal muscular atrophy gene therapy. But it seems that at the moment they are not taking the risk yet, and we are still early stage. I think for them, if we could show that these linkers are not toxic, that we are basically having readouts to actually run clinical trials, we are hopeful that then they would eventually take over.
So complex equation. Any other comments on the clinical? Anne?
I just want to say, regarding gene therapy, I am still extraordinarily optimistic, and I've been active with both clinical and research gene therapy for now 7.5 years. And I do think that the lessons of gene therapy for treating spinal muscular atrophy are different than the lessons of treating muscle. And I think it can be done safely, and I think that we're still collectively around the country and around the world learning some lessons about that. So I just want to put out there that I am still extraordinarily optimistic.
Yeah, I would think so too. I think it's all. There were some tragic news, of course, in gene therapy in the Duchenne trials and limb girdle and myotubular myopathy trials. But every single case, as tragic as it is, teaches us lessons how to move forward, as long as everyone is transparent about what happens. And so that's something that, Anne and I and others are very passionate about, to really ask people to come to the table. Also, the developers, the sponsors, industry that puts it into patients, to say, well, this is, no one wants to blame you, but you have to put the data on the table so we can understand what happens and how to prevent it and make gene therapy safer. And that's an approach that has worked reasonably well, and we've gotten actually quite a bit of buy-in to do that. And certain pictures emerges around the liver toxicity, and it is really a function also of the total amount of virus you have to give to a person to treat the muscle. And most of these events happened with capsids, so with virus capsids that were found in nature. So those are basically things that you find and then re-engineer to deliver the tools. But the field has moved on now in developing viral capsids that are much more attuned. They're engineered to go less to the liver and better to the muscle. So they're kind of called myo-AAV and AAV-myo, and that allows you to pass through the liver more safely. and go to the muscle more effectively. And that allows you to then lower the dose, increasing the safety margin of what you do. And so I think there are ways to really scientifically understand the problem and engineer your way through that to a safer gene therapy. I agree, we're in the early stages of gene therapy. When it works, it is astounding. So the process, the promise of it is right there. But we have to understand the safety margins of it and how to improve it and what to do across the entire field to make this an attainable and a safer treatment for everyone. So and this is really the transparency and data sharing, but the other aspect of genetic therapies really that's important to understand is that these therapies are not, of course what you deliver is disease specific, but what you do is not. So you're basically using a platform, how you do gene therapy, that you can compare across different diseases. So that means that the entire field of genetic medicines learns as a field about these platforms, so that things don't happen to have over and over again in every single disease. But once it happens in one disease, others can learn from it and do it better. So that's an aspect that I think propels rare disease therapeutics by understanding the platform nature of it and how everyone can learn from everyone else, as long as there's transparency and data sharing, something that we as academicians, you as patient organizations can demand. You can say, I'm going to participate in this research as long as the data is shared and made available for everyone else. I don't know whether anyone has comments on this. I'm passionate about this, as you can tell, but any?
Yeah, I just completely, support exactly what you said. I think transparency and, I think there was some hope that, as I say, just thinking about muscle delivery separately, I think some of the lessons that were learned in the early goings at 1 or 2 sites did not translate to 15, 20, 40, 50, 60 sites around the country and around the world. And so I just do think that transparency in science is critical, and I think families deserve that same information, and I think that's becoming more and more the norm.
Yeah, I think the question was how much time it will take for treatment to develop. I think it's also a bit dependent on the genetic cause of the disease. If you, for example, these splice site modulators can use, they're already 18 in the clinic, and but that depends really on what is the genetic basis of the disease in your child, and that can also change the way how the perspective is. And the other things of all this therapy, I think the real issue is also not only the effectiveness, but also the cost. How much will it going to cost? And if we did talk about 10 million or 20 million in trials of investors who have to get a profit of 2-3 times, then you might end up in a problematic situation if you have a rare disease. And that is why also with these stem cells, we try to keep it also in a hospital environment. That will not be possible. I just talked about it with Markus for the viruses. But if you can have a hospital exemption, if hospital clinics can raise these stem cells, that might be a way of keeping the cost down. You do not need the investors then, as long as these prices are being covered by the health insurance. In the US, it might be different than in the Netherlands. I'm not sure about these things, but there is really a keen role on how to reduce the cost, keep the costs low of these treatments. And I think the patients can play a very critical role in that, in making the awareness to the regulatory agency that rare diseases are worth a treatment, but at a fair price. And that is what is, I think, also a key issue.
I don't know, this is just me thinking outside the box. As my kids get older into their adulthood, like, we're pretty used to living with this disease now, but one thing that as they're getting older and you fear, obviously the worst, just something to help them be stronger. I'm not looking for a cure, obviously the cure would be the ultimate goal, And I don't know, like Spinraza for SMA, and I don't know if that's a form of gene therapy or not, or just is it a form of gene therapy? Okay, I wasn't sure if it was just a medication or not. Is there anything, are you working on anything just to help quality of life? Not necessarily a cure, but just quality of life.
So one of the interests we have is looking at muscle growth. We know that in LAMA2 CMD, muscle growth is disrupted. Sorry?
Which agent?
Oh, just looking at muscle growth, just the ability of muscle to grow. So something very cool about fish is that they can make new muscle throughout their life. And that's the concept there, that can we, as you said, it may not be a cure, but can we improve the growth of muscle? And that in itself could be beneficial. So that's something, again, at super early stages. I don't have any compound names or mechanisms. I mean, there's some data out there with the IGF-1 pathway that if you can improve muscle growth, that is beneficial. Yeah, it's not a cure. But, you know, looking at those sort of mechanisms, yes, they're on the pipeline, and we're looking at that too.
Yeah, I think it's a fair point you raised. We had a student in our lab who was also LAMA2 patient of the age of your sons, and he was also working on the disease himself, but he was also very keen on what he would like to achieve as a bit of a treatment which would increase in independency. It's not so much a complete cure. And that is, I think, we should learn from you what should be the targets we are addressing, and maybe some local benefits might already be good enough and might not be the full cure, but are really very helpful in the stage you are. Yeah.
Can I just add that also from that perspective, I mean, we talk about gene therapy, and it's absolutely super exciting, but there's drug repurposing as well. There's so much we know about all these chemicals that are approved, FDA approved, getting used for other diseases. Just a concept, can we try and use those and try and improve, you know, it will speed up the whole process, because, yeah, it's a time gene therapy or any other new chemical, it's you know, getting it through the whole pipeline, it takes years. But yeah, just a concept of repurposing and then using combination therapies as well, which is, again, something that I'm very interested in.
And maybe, Veronica, how does your research factor into that? Do you expect to find things that you can...
Yeah, now our research started from the identification of one family in which two siblings with the complete loss of laminin have different disease severity. So basically we studied the DNA and the muscle gene expression of the genes expressed only in these patients, which is as an atypical manifestation. We then tend to identify some genes that we can modify in order to ameliorate the disease severity. At the moment, we identify a few, and we tested some drugs that were already available in animal models. The first one that we tested, unfortunately, didn't work, but we're still on the search for other drugs, and eventually we hope that even though we know that we won't cure a disease in itself because we're not targeting the defective gene, in this case laminin, but starting from the observation of this patient, which has an atypical disease and a milder disease, if we found a gene that can actually ameliorate the disease severity in this patient, we can actually improve disease severity and consequently also improve quality of life.
Yeah, and that's really important, but what I mentioned before, this multimodality of therapies, that even in the best of circumstances, the gene therapy will not create a new muscle that is disease-free out of the blue. So it's very important to kind of use any angle that you have at the disease to make the muscle more resistant to it and help it to recover. And that would, I think, in my opinion, apply at any age and in any combination. One of the challenges, though, and I can ask the panel for opinions, or anyone in the audience for that matter, is how do you do this type of drug development in an ultra-rare disease population? Because, you know, if you go by the books, you have to do every single intervention, even if it's a repurposed medication, in its own right as a clinical trial. And we don't probably have the bandwidth, the funding, or the resources to do this for every single one of these repurposed drugs. So we have to be creative of how we do this without kind of losing against time to do that. I welcome ideas. Okay, then I'll leave it out there. And
I have a question on, excuse me, funding, right? The work you all are doing relies heavily on the funding that is available and made available to you all to do the work. And whether those funds come from a private donor or not, this isn't a question about policy or the United States government. When funding is removed or changed abruptly, I imagine that has implications on the work you're doing. Can you help us understand the practical impact near term and long term with respect to funding? And when funding sources dry up, again, whether it's from a private donor or from a government agency, what does that mean to you all today? What would it mean to this group in 5 years, and just the prognosis of everything, and ultimately trying to find a cure or increase quality of life?
Well, that's a great question. You're on the spot, all of you.
Oh, it's a great question, and I think research is not a start-stop button, right? Funding dries up. It doesn't mean you stop there and then you get funding the year later. It means you start back. There's actually a startup period, and that's where the delay comes in. For me, I'm quite a new group leader, and I think one of the biggest challenges is the small pots of money that come through. You're just barely surviving to make a change, and all of a sudden that dries up, and it's it really does delay progress. One thing that I do want to say, and especially in this crowd and in the Australian system, it's worked really, really well, is advocacy. I, as a researcher, go to the government and tell them, hey, I need money. They'll just laugh me off and say, you're whining, and that's what we researchers do. What's really has happened is, especially in the congenital muscular space, the advocacy people have gone to the government and said, we want more money. 2 years later, there's this huge funding call that came from the government. In fact, we were successful with it. The point there is, yes, the loss of funding really does delay us. It's not just, it doesn't mean we start where we left off. Actually, we've got to start from the beginning. So funding needs to be continual, and you can't just stop it. But the second thing is the role that you guys play. You've got so much power. I cannot explain it to you, because I've personally seen that happen in the Australian space. So if you want change, you're going to have to drive it from your end, and this open communication is what's going to work in that space.
Can I just add a comment? I was at this conference in Chicago, I don't know, was it 6 years ago maybe, and it's when I met Carsten and Reghan and Veronica. I remember I had a conversation with you at the time, and I took a lot back from that back home. And I'm naturally one, and my wife and I are naturally ones to tell our son's story and do it in a way, we need people to get to know my son, right? And to care for him and love him, but also to do something on his behalf, right? And we talk about advocacy and getting people to work for you. And I find it effective to, again, tell my son's story and just help people understand the importance of advocating. And I found that, there's a lot of people out there that are willing to do that on our behalfs. But my point is, I remember I took something back from our conversation in Chicago, and I'm in Nashville as well a few years ago, and I know this year. And the work you guys are doing is incredibly important. And speaking for myself, I take a lot out of these, and I bring it home with me, and put it into into my work at home, and just trying to advocate for my son, for this community on behalf. So just, I guess, a thank you for all the work that you all are doing. And just know that me, my wife, and I'm sure everyone else in this room, we're doing our best at home to try to make sure people are paying attention and helping out here.
Well, thank you, and I have some comments on that too, but Bert wants to say something.
Yeah, I think getting funding is the story of the life of a scientist, I must say, in any situation. And for me, it's also spreading the risk. We have about, if you see my slide on the meeting on Tuesday, we have about 20 organizations that fund us. And that varies from muscle foundations, patient organizations, basic science research, translational research. And that's our obligation to be creative in doing that. But I must say also the patient organizations really make a difference, because we have the Foundation for Sarah in the Netherlands. The founder is Bram. He's a journalist. He does our PR. He's so visible. That attracts a lot of money, and that is also money for us for this research. And that helps also bridging the gaps, which were mentioned a moment ago. You're not always lucky every year. There are years when you do well, there are years when you do less, and then these make really the difference of being able to continue the research at the level you need. So the patient organizations do have a very important role there, not in the amount, but in making things going on.
Yeah, but I think we have to acknowledge that it is a challenging time at the moment. I think it would be. We're
from Europe.
Yes. and it gets challenging when science, medicine, and the needs of rare disease and patients become political. You know, that's when it gets very challenging and becomes a tool for political purposes. And but there's a hopeful sign here too. So in last week, in the Senate Appropriations Committee that decides on funding and grants, there was a surprisingly strong bipartisan agreement that NIH funding should not be cut and increased by a certain amount even. So in that kind of committee where people are really, you know, are put on the spot and their beliefs, there is bipartisan understanding that supporting health science research is something that is extremely important and cannot be cut. What also means is that there is that understanding that sometimes people may no longer feel comfortable to kind of say publicly that this is important and needs to be supported. But there will be more comfortable saying that if they hear from, I think, the constituency that that is what they expect. And so if you create that courage to stand by this, then I think you'll have more converts there and a larger, you know, a larger dam against unnecessary cuts. And I hope that'll be the theme of the coming years, that there will be some kind of defense that protects funding. But, you know, it's your dollars, it's your children, and I think you have the right to ask for that.
Yeah, Dr. Bonnemann, there's been obviously news about funding, but there's also been news about maybe the FDA accelerating cures for rare disease with the right proven vehicle. Is there much changing there that is hopeful?
So yeah, that FDA at the moment is also very unsettled. You know, Peter Mark's successor didn't last very long, and so we have to see what comes next. So I hope people stick to, you know, what's really important, which is developing therapies and cures, and leave everything else for others to quibble about. Yeah. So, yeah.
That was actually one of my questions, so thanks for asking that. It seems silly to ask any questions. I want to go home and start campaigning, like right now. So I have a couple questions. I think I got the gist, like for stem cell therapy, this is something that we derive in a lab. This isn't something that's a donor. I think that was answered. This is something that they're deriving in a lab. And if you could, again, I've heard this many times, gene editing and gene therapy, like what the difference is, and are we talking about gene editing at all, or is this all gene therapy? We could stop there.
Yeah. Shall we start with those two? Stem cells goes to Bert.
Sorry, I was looking in the light. Can you please?
Oh, over here.
Yeah, sure. What was your exact question? Because it was.
Are stem cells, is this something that we derive in a lab, or is this something that we are using as a donor?
No, this is autologous stem cells, so it's patient-derived cells we use.
Okay.
And we culture them in a GMP facility because you have to bring them back into the patient, so they have to be cultured in all kind of regulations that it's safe to bring them back. And we're currently proceeding already in clinical trials. We have tested safety, which is good, and we administer them through the arteries, which go through the muscles. So it's systemic treatment, but and we're now looking for efficacy. And then indeed, I think there needs to be some improvements, but we do it first locally to be really sure about the endpoints, because that's also something you really want to measure the effect. And I think, yeah, cost-wise, it's in the cost of other stem cell treatments you might be familiar with, so it should be doable.
Oh, cool.
And the correction, indeed, yeah, that is, we want to correct these adult stem cells, which is more difficult than iPSC type of stem cells. But I must say, there's now been new culture media developed, which all these adult stem cells can grow indefinitely with better nuclear quality, which also opens up new opportunities to use them in the future. Yeah.
Wow, that's cool.
Okay, so a little primer on gene therapy and gene editing. Gene therapy, in the current use of the word, is AAV-mediated gene therapy, so we talked a lot about that here. And what that means is, as I mentioned before, you have a viral capsid, a viral envelope that delivers a small piece of DNA that has corrective action. And it gets to cells reasonably well. It gets to the cell nucleus where the genes live, but it does not integrate into the chromosomes. So it stays basically as its own little piece of DNA in the cell nucleus and does not integrate into the genetic material of the patient, which, is a good thing because you don't randomly kind of destroy things in the chromosomes there. And that's a good thing until the cell decides to divide. When the cell starts to divide, it basically divvies up its genetic information by replicating the genetic information, the chromosomes, so that each daughter cell gets the same amount of genetic information. And that's where the AAV DNA is left behind, because that can't replicate. It basically gets diluted out as the cells divide. And that is in neurological disease, where nerve cells never divide anymore, is not an issue. Once you get it into a nerve cell, it stays there for as long as we hope it will. But if you're in a muscle cell where you treat a young baby with a muscle that grows, a muscle that turns over because it tries to repair itself, that actually is an issue, because in that tissue, you're losing the genetic information, the gene therapy over time as the muscle grows and adds new cell nuclei to it and repairs itself. So we can maybe get some opinions on that later. So that means you have to refresh the gene therapy as the child grows. And that's where we're really bad. We can't do that at the moment because the immune system now knows your viral vector and will reject it next time you try to sneak it in. So that's where gene editing comes in. If you edit it in the chromosomes, then your repair, your edit will be inherited as the cells proliferate. So as the cells divide, now all the daughter cells have it and actually amplifies the effect of your therapy because all the new cells will have the genetic edit, the correction in it. Problem is, it is at the moment highly inefficient to do that. And secondly, the safety margin of that hasn't been established because you're really now cutting in the genetic information itself. And you cannot afford errors. You have to be highly precise and only do effectively what you want it to do. So you've heard, and then you have to deliver it to all cells where you want to go. But you've heard about this amazing case here in Philadelphia of this young baby with the metabolic disease where the gene editing was given to the liver, and the liver cells were edited and corrected, and so far so good. So that it tells you it can, in principle, be done if you effectively and safely deliver it. We haven't talked about it, but delivering to the liver is actually fairly trivial because everything that you give into the vein goes to the liver and mostly stays there. But to go body-wide to the muscle is a real challenge that we have to address. But gene editing is also an option for the stem cells, of course, because in the stem cells you can do it in a very controlled fashion, and you only take the stem cells that are properly and safely corrected, and that actually is FDA approved in the clinic already for blood disorders. So we're making progress there, but we have to work with what at the moment has the highest chance of changing the disease, and that is AAV-mediated gene therapy. Doesn't mean that the other things shouldn't develop at the same time, but we have to kind of start with something that is closer to the clinic while we develop other things. I don't know whether other people want to comment.
Yeah, I maybe also comment on gene editing is usually understood as you correct the specific mutation of these patients. Mm-hmm. And with LAMA2, we know that almost each patient has a different mutations. And so it's already difficult to develop therapy for this rare disease. So that's why most of the therapies we are working on should work mutation independent. So we try to develop an approach which work independent of the mutation. And so for gene editing, so to correct the mutation, you would almost need to develop a therapy individually for each patient,
that's what they
which
did
is
in
an
the little
additional
baby.
challenge.
That's what they did in the little baby, specifically for that.
Thank you.
Yeah.
I did have one more question. So I know we're talking a lot about muscle integrity, which was already resonated, like if we could just gain some more muscle or maintain what we have, that's amazing. But with LAMA2, you know, there's the white matter changes in the brain, and I don't know if you guys clinically see changes like, oh, if your MRI looks like this, they might progress more quickly or anything like that. So just clinically, does that ever change things? And are any of these treatments going to change like neurologic changes?
Okay, anyone want to answer? Clara? Anne?
Apparently, those patients, they do not worsen on the neurological part because the brain MRI seems to be stable, but we're looking into that, so we can be sure to answer that to you. But as far as I know, these kind of gene therapies don't reach the central nervous system, so I don't think that's going to be the main goal of them.
Yeah, the white matter disease really is not a breakdown of the white matter. It really is a reflection of increased water content in the white matter that shows up. But there is, in some children, a substantial proportion, maybe 15 to 20 to 30%, have differences how the brain is formed during development in the back of the brain, which isn't a big issue for the children because they remap everything from these regions, but it can be an issue for causing seizures to emerge from these regions. And that would not be expected to be addressed by the gene therapy for two reasons. One is that's differently formed brain that you cannot correct, and secondly, you don't quite get there. But the other issue, of course, is the peripheral nerve, the motor nerve that go from the spinal cord to the muscles, and that's a big deal in the mouse, and we don't quite know how big a deal that is in the patients, but can you correct that with a gene therapy?
So this is actually also ongoing discussions between the mouse researchers and the clinicians, because in mice we realized we can treat the muscle and the muscle looks better, but then we see very strongly this problem in the nerve. And so we discussed this with clinicians, and they tell us it's stronger in the mice than it is in the patients. But in the mice, we have tested if we can also treat this peripheral neuropathy, and we have modified our therapy in a way that we can. But then it's also now a discussion in terms of safety and so on, if we should try, is this the way to try in patients, including the nerve, yes or no. So
if you were to perfectly treat the muscle and you don't treat the nerve, would that be a problem later on in the life of a child with LAMA2? Anne, any clinicians you want to give?
Children with LAMA2 have normal sensory function, and we test that in some simple ways. We ask them, do you feel light touch? And we compare up to down, and it's the same. Then we go another step. We say, well, how about, the scratch of a pin? That's a slightly different type of fibers, lots of different sensory fibers coming north. And we do that, and they nope, that's the same. And so then we pretty OCD in the neurology world, so we check up and down. Can you tell the difference between up and down? That's a whole different set of fibers, and they seem to be able to detect those too. That said, there was clearly peripheral nerve loss of LAMA2. If you actually take a little nerve biopsy, somebody did that many years ago, and it is missing from nerve. The other kind of funny thing about nerve is there's actually motor nerves, of course, within your muscle fibers. So could it mean that you might, on a bigger level, not just the sensation, but the muscle fibers within the muscle, could those be affected? Or could those be involved that we can't really see it? It's silent because they're weak. And I suppose that's theoretically possible, but as best we can tell as clinicians.
Yeah. I mean, we as clinicians have to study this better, I think, and more proactively. And secondly, probably if we can safely also get it to the nerve, that can't hurt, can it? And so can you do that, Markus?
Should I answer? So it's, in terms of safety, this is so like as Carsten has explained, I think so central nervous system versus peripheral nervous system. And we know that in LAMA2 MD, the brain shows some white matter abnormalities, but we also, in terms of safety, we think we should maybe not bring a virus into the brain when we don't have to, because it also brings an additional risk that you change something in the brain which we don't want to risk. So there are AAVs available which actually go to nerve and muscle, and they don't go to the brain, and they work in mice. So in mice we can do it. If it's safely possible in humans, has to be evaluated.
Yeah, I should maybe also say at the moment our gene therapy is based on AAVs that are already in clinical trials. We are not using any of these experimental myo targeting and muscle targeting AAVs, because we also want to target actually the peripheral nerve, with the hope that having these AAVs that are safe in humans, that they are actually also faster to eventually a clinical trial. That's the idea, because these new capsids, you would need to test them in toxicology and all kinds of things. And I mean, we are not in with this rare disease. This would be an additional burden and slowdown and money requirement that we can't really do.
Yeah, that's good point. And not every capsid is created equally, and so they all have their different tox profile. And I hope we learn as we move closer to clinical translation enough so that we can do this safely, which is my belief we can. But let me challenge you here on a point that's also up here, which is when is the optimal time to treat? What improvement can we expect? Is there a closing window of opportunity in gene therapy for this condition? We know all that in spinal muscular atrophy, a neurological condition, that you have a very narrow window to be effective. You really literally have to come in before the disease is even fully manifest to completely correct it. That's a nerve disease where the nerve doesn't regenerate or repair. Here we have a disease that is clearly already in ongoing mode of regeneration when we diagnose it, but it continues to do so. So do we have any idea clinically from the preclinical work whether later in the treatment is still possible? What is that window of opportunity? What can we expect?
Yeah, so with our approach, I think in mice also we see the disease is similar as in the early onset patient. It's already present at birth, and then it progresses very fast. And in the mice, we have tested both, just an application of the gene therapy directly after birth or later. And we still see significant treatment effects when we apply it later, but the treatment effects are bigger when you apply it earlier. I think this is just in the sense of, in general, when a lot of muscle is already lost, then muscle, in contrast to nerves, can regenerate. So you can actually build muscles again when they are lost, but there is a limitation of this. So when a lot of muscle is already lost, you can eventually stabilize them to not more degenerate, but you cannot then suddenly build up very strong and very big muscles.
Yeah, I would say I definitely followed that, followed your work on that. I was very excited about that because one thing that is very different about LAMA2, and most of you have older children know this, hand function, biceps function, yes, there are contractures, but function, the ability to feed yourself, the ability to, straighten your glasses, or, those motor functions do not usually completely go away, maybe even for very, 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 very long periods of time, maybe ever. But so there is something very different about LAMA2 where there may still be an opportunity. It's very different than Duchenne, where ultimately a man with Duchenne loses every bit of hand function eventually. But so I don't think I think that window may be bigger in LAMA2 than it is for more progressive dystrophies like Duchenne.
Interesting thought. Yeah.
because they get bigger, like holding themselves up, and probably their spinal fusion. But otherwise, a lot of things have stayed pretty stable.
Pretty
Like
stable, yeah.
It's been pretty much
No,
the same. their whole
exactly, and I think so. There is, I think, a reason to be optimistic that there can be some kind of repair and turnover of tissue later. So one of the things that you also see quite prominently in a muscle of a child with LAMA2 or young adult with LAMA2 is the buildup of connective tissue in the muscle, the fibrosis in the muscle. And that until very recently was said to be kind of a point of no return. Once you have that there, that stays there. But if you look a little bit left field into other conditions where fibrosis is happening, like liver fibrosis, kidney fibrosis, lung fibrosis, it actually turns out that fibrosis is much more dynamic than we thought it is. There is much more kind of turnover in the tissue that's happening. And so that raises the prospect that if you fundamentally change what's happening in this disease and give it the chance to remodel, that some of that could happen. I wonder what you guys think.
I mean, when we say we treat later in disease, it's a 3-week-old mouse, which corresponds to a 10-year-old child approximately, almost ready to be fertile. And this still works. And these mice are really heavily affected already at birth. And I mean, they grow. And we haven't tested how long it's going to last, but we see a clear benefit when we do this and we treat these animals.
Okay. There was another, Sandra, who you're pointing to?
It's all good. Yeah. So please forgive me if this was already answered, but I don't have a scientific mind. So just like for like real life lay parents in this, should we expect that with these therapies that may come about, that there would be improvement in that they would be able to do things that they have not already been able to do, like lift their arms over their heads, weight bear in the legs, something like that? Or are we just to expect it not to get worse than it is now?
Well, thanks for asking that. That's actually a very, very good and very scientific question, because it plays into how we think about clinical trials. What do we want to show? Do we just want to show that things are not getting worse, are getting worse more slowly, or do we really want to show an improvement? And I go out on a limb here, and I'm going to say we want to see an improvement. We want to see the muscle get better. And the reason why I'm saying that is because, as you heard, there is this potential of muscle to regenerate and build and rebuild. And we have actually, this is a completely different disease, but it's also a congenital muscle disease, X-linked myotubular myopathy, where gene therapy was done. This is, of course, a different disease, not a dystrophy, but also a disease with very, very abnormal muscle at the get-go. And the gene therapy in that trial in children who were not even able to sit or be on breathing on their own resulted in remarkable improvement in some of the children to standing and getting off the respirator, telling you if you give muscle what it is lacking, then it will try to kick in and repair. Again, saying this is a completely different muscle disease, but that a potential of muscle to heal is there, and gives me the hope that the answer is we're looking for improvement. I don't know what others think.
I had a follow-up question to that with, like, as far as, like, orthopedics. I didn't go to the orthopedic lecture, but, like, my son is at the point where they said he needs to have surgery to correct his hip dysplasia, like the heads of the femur coming out of the acetabulum. So this is a really good question. I'm glad you brought that up, because, I mean, my expectation is he's not going to weight bear and ambulate on his feet, which is fine. We're, good with that. But, like, would we want to think differently knowing that these therapies could be coming?
Well, I was planning to comment on the previous one. I think what was mentioned stresses the point Carsten made on the multimodal therapy, that you would have gene therapy combined with cell therapy for the regeneration, antifibrotic therapies, and then I think you can reach improvements. But that requires a multimodal approach. Yeah.
No, exactly. To your question, that's a really good one, and My personal feeling is, and again, I'm very happy to take fire for that, but is to stick to standards of care, not expecting the therapy to be a miracle cure. And so kind of do what's best for the patient at this point in time in your orthopedic management. And then if there is a therapy that makes it substantially better, react to that and when it comes. But not delay or anticipate therapies because of that. That would be, I see some nodding from clinicians, or okay, good.
It is important, especially for hip dysplasia. Hip quote-unquote dysplasia is really not dysplasia. It's just that the normal weight bearing that a child has in order to form the joint around the acetabulum does not form because they cannot weight bear. So you can imagine trying to fix that. They're still not weight bearing for the next year or so, and so it's not a trivial surgery. It is painful, and so for that particular surgery, we've typically said unless the child is in pain, it may not be the right approach. Children who have hip dysplasia and normal strength, they get put in these lovely little braces that sort of force their hips to reform that joint. But I would sort of second the common sense that Carsten put forth. And I've seen enthusiastic, usually very young orthopedic surgeons say, well, we should fix this now because we can't fix it later. But again, the child is having they cannot fix it in a way that the child will continue to grow and continue to fix it themselves.
Great. With that, we are at the end of our time, and I'd like to thank the entire panel and also the audience for participating in this session. Oh, there's one more question. I'm sorry.
I'm sorry.
Yeah.
This is related, but a little bit probably off topic. The background is we have a sibling who is a carrier, had a major cardiac event at a very young age that has no explanation to this day. And I'm just curious if, I know with it being such a rare condition that we're kind of just gathering information from the affected individuals, but I'm kind of curious if there's been any research done for carrier status. And the reason I ask is because I know that there's also a parent in Germany who also suffered from a cardiac event who obviously is a carrier. So I guess with no medical explanation that could be found, we're just curious.
Well, it's a super question. Who wants to comment on that?
I'll take a crack at it. So I think that from other and published stories of other muscular dystrophies, there has been disproportionate involvement of one organ system versus another. What we believe is that, and specifically, one child got a heart transplant, and the sibling was still running, and they had the same genetic disease. One got a heart transplant at 8 months, and the other one was still running at 10 with normal heart function. Having said that, the other cardiac events, there's something in medicine we call double trouble. So for your second child, I would have all hands on deck to think about, it's possible one child had something and the other child had something quite different. And so doing the full genetic.
Sorry, I guess maybe I stated this wrong. It was one of our siblings. So one of our siblings is a carrier of this same as us.
So the next generation up.
Yes. Okay. So not our children.
Oh, okay. Okay.
So we have an affected child, but we have a sibling that had a major cardiac event before the age of 40 with no medical explanation of what happened. And I know that there can be cardiac involvement with our affected child. So I'm wondering, has there been any studies for carrier status? And if we could also end up experiencing cardiac events?
Yeah, so your sibling was confirmed carrier?
Yes.
Okay. That's a good question, and so they maybe I should kind of say it in two ways. One is so far no, that hasn't emerged from just looking at big cohorts of families. But on the other hand, I have to also say that there haven't been deeply phenotyping studies of carriers to really look with cardiac imaging and EKGs into all of these. My gut feeling would be that it is highly unlikely to be a cardiac risk in its own right, unless something else comes in addition to that, be it a genetic risk factor or something else. So I think that may have to be explored further. I know of no data to show that just carrier status it causes any increased cardiac risk. Even in the children who have completely LAMA2 deficient, the cardiac events are still rare and often just subclinical. So I would be surprised if carrier status alone would be enough to cause a cardiac risk. But I wonder what others think. Yeah. But thanks for the question. I think that something we should bring back to the consortia to really take that very carefully in the family history to look exactly for that in families and investigate it.